Okay. Hey, welcome, Michael. Don't know if you're all the way in the room yet. Okay. Okay, now he's in. Hey, Michael, good to see you. Or at least your picture. Okay, so we just started our recording. We'll be
Hey there,
letting.
John.
Hi, how are you?
Good.
Are you in the plant or is that your background?
That's a fake background of Merck Durham Technical Operations, which we used to support back when.
All right. Very impressive. Okay, we're going to get started. People will be coming in and going out from time to time. We have a big array of panelists to answer any stability questions. And we have some questions that have been submitted in advance. And I'll introduce our panelists. This is alphabetical order. Michael Brommer, sales manager from Parameter Generation and Control. They make excellent stability chambers. Jeff Carr, retired director, analytical development, Thermo Fisher Scientific and Patheon, who has just joined us as a StabilityHub contributing correspondent for North America. Bashar Deeb, quality manager from Precision Stability Storage, and he has facilities in California, Florida, and Massachusetts. Maria Krish, research fellow, physical sciences from Freethink Technologies. Chris Latos is manager of stability services at Hollister. Myself, former associate director for QC stability at Regeneron, and a longtime PSDG facilitator and now editor at StabilityHub. Walter Ralph, retired director of global stability and reference standards from AbbVie, who's our associate editor at StabilityHub. Stephen Schwartz, director of business development, East Division from LAB, and he's expert in validation and monitoring solutions. Amy Wong, associate director from Arcutis Biotherapeutics. And for our technical support and administrative support, Christina O'Neill, and she is a relative, full disclosure, former executive assistant at Sanofi and coordinator for PSDG events. So that's who you're looking at the moment, plus our guest. So we'll start off. We know, Amy, your time is limited. I'm not sure how many people are into therapeutics, but we're recording that right now. So can you give us a little bit of idea about the hot topics there are for therapeutic stability? We find it fascinating that we know it's very short term and requires super duper cold temperatures, but we want to be knowledgeable about that because it's among the fastest growing areas in the stability world.
So I just wanted to understand, so therapeutics meaning more like the biologics, proteins, peptides, cell and gene therapy, everything like that. So that's kind of all encompassing, right? Therapeutics.
Yeah, and you can get specific in any one area that you feel comfortable with. Just tell us which one you want to focus on.
I don't think I'm going to focus on any one in particular. However, I've done a lot of protein peptides and I've done some cell and gene therapy work. So the things that matter to us obviously is you have something that doesn't necessarily do well in room temperature for the most part. And so your target long-term storage is, you're probably going to want to look for refrigerated as opposed to frozen if at all possible, but it might not be possible. And so you kind of you do your studies in a manner which you try to broadly capture everything, like you can start from refrigerated and then go to different frozen temperatures, and then as well as doing accelerated studies at like maybe 25 degrees or something like that, and maybe even 40 degrees. It just kind of depends on what your molecule can withstand. The higher temperatures, the more propensity you get for gelling and precipitation and everything, and so that you may have to stop your studies early if it gets too warm, too hot, so that you totally degrade your molecule. So it's kind of like you just have to just broadly study a lot of different things up front. So pre-formulation is very important. We generally start with a lot of pre-formulation work and many different formulations, screening formulations, and then from there pick the lead formulation, and then go on to methods development, and developing stability indicating methods. And that is also very difficult too, because there's, larger molecules, a lot more to look at. And so methods, starting with like HPLC methods, reverse phase, and then you might want to do isoelectric focusing, might want to do size, size exclusion, lots of different methods. Something to look at would be starting with the USP. USP1, I think, is it for injectables, for general injectables. But I think there's one USP, I think it's 1041, and that's particular for biologics. So there would give you some guidance. And of course, every biologic is probably very unique. And so a lot of the methods development is in-house. There may not be a lot of things pertaining to your molecule in the compendia. And so some of the hot topics that I remember working on is particle size. That's always, when you're talking about the subvisible and the visible particles. I think it's 788 is for general particle sizing, but 787, there's 787 for biologics right now. That has always been the challenge for us. I think it's it's sort of a new field. There's a lot more technology now. There's a lot of technology flooding the market to try to help capture how to do particle analysis for biologics, especially because for biologics, there's not a lot of material. So usually your formulations are very limited. Your dosage form may be limited as well. And so you don't want to quote unquote quote waste a lot of material doing particle sizing. And so so USP 787 allows for you to develop your own method, kind of based on, the Hayek method and things like that, but it allows you to diverge and, come up with your own, and then you just have to justify everything. So that's better now, because you can develop your method, based on the sample size that you have available for your drugs, your molecules. So those are some of the hot topics I remembered. It's been a while since I've worked on those, but those are the hot topics that I remembered.
Let's pause for a second and ask if any of the audience has any questions in this particular area. If not, we can continue on with a question or two from Amy. Either use the chat or just raise your hand. And your raise their hand features you'll find at the bottom of your screen. If they've all disappeared on you, you can click on your screen or touch your screen and they'll all come back. Then hover over something. So we're watching for those questions to come through. If you're thinking of any at the moment, Stan, hey, I can also unmute and just speak up if you like to.
I have one.
Sure.
So colleague posed a question in the context of monoclonal antibodies and, if you will, matrixing, which was that if you're trying to compete with a biosimilar to market and you are trying to get an advantage, normally you would have a BLA submitted with 3 PPQ lots. Everything's been on stability, 6 months accelerated, 12 months at the intended 2 to 8 storage condition. And we were discussing, could we potentially file 6 on just the 6-month time point? And some of the avenues we were discussing were, well, if we potentially had a rolling submission that was going to happen over 6 months, perhaps we could get CDER to agree that we would be submitting initially with a 6-month on the 3rd lot. and then during the rolling submission time period, get them the 9-month, and in what circumstances they might be willing to allow that approach. But of course, CEDAR looks at things from a different standpoint, which is your hurry up and your need to get to market is not necessarily as important as our first time right on reviewing. So have you seen some examples where perhaps by using the comparator molecule, or maybe in a situation where you have a prophylactic mAb and there's 8 or even 12 mAbs in the drug product, that you might be able to do some matrixing and have not quite all 12 months of data ready for the BLA?
I think you managed to give us the most complicated question of the day, Michael. Thank you.
Yeah, it's obviously a DoD project from once when, and it's about how difficult it is to produce first 8 antibodies for botulism, and then they decided, no, let's see what the 16 will look like. And it's a stability challenge, not just the timing of the BLA, but also on the poor QC lab on managing and pulling all those samples, getting them on test in a timely manner.
We'll ask Amy if she had any experience or impressions on that. That's a big one, or if anybody else wants to chime in right after that. If I know FreeThink has been asked about biologics, but I'm not sure that you've got an application there. But we'll turn to Maria after we check with Amy.
That's a great question. It's a very loaded question. I don't have I don't have the experience doing that, so we haven't tried that. I mean, most places that I have been at, we've waited for the 12 months long-term stability. But I think that if, in your situation, it might be a good idea to maybe ask that ask that of the FDA in your pre-submission meetings. You may never know. I mean, they probably won't like it, but you could probably file it, and then, like you say, just it's a kind of a rolling thing. By the time they review it and ask, then you'd have the last 12 months. But it is a risk. It's a big risk. I don't know.
Okay, that's fair enough. It's complex, and I think I've seen similar things actually work, but it's kind of case by case, as you mentioned. It has to go through the FDA and check. Anybody else want to comment on there if you had experience on it, or you see something very similar that either worked or didn't work?
My only comment was just, I believe I heard you mentioning matrixing, and it sounded like you were implementing matrixing within those 3 studies, which it sounds very unlikely you would have nearly sufficient data at 6 months if you were doing that.
Right. So the reason why matrixing came up was because each mAb is produced with the same process in the same suite, side by side, 300-liter bioreactors, same staff, same facility. So at least from the manufacturing consistency standpoint, there's substantial comparability. But of course, the molecule itself has all the expected differences in glycosylation, and then how those glycans degrade during the protocol.
Maria?
So you had mentioned FreeThink a little bit. So I work in a company that does both provide software and does studies of sort of accelerated stability models. So mAbs are on the outer size limit of where people tend to use these. It gets a little bit more case by case as you get bigger in terms of how well the models approve. Some cases it's worked very well. But just in general, from a regulatory point of view, a lot of companies that do, especially our software users, do use that approach to kind of speed the time to clinic. So they include, we don't do the filings ourselves, but just from what we've heard, they do include a stability model and then a smaller amount of data that agrees well with that stability model, and then sort of continuing testing. So that's not uncommon to do for smaller systems. those models have been used for larger systems, less common, and it does depend a little bit on what's limiting. But that is one option that I think is one of the most common uses of these kinds of predictive models, is just to get into the clinic with continued testing on sort of a, a smaller time data set that then is continued. So.
I would say for larger molecules, there's we've had experience where bracketing is also already frowned upon. So matrixing, especially with something that is that complex, it's not, I would say it's not likely that they would approve it. Let's just say, for example, I've worked on something that, same molecule, same formulation, it's just a concentration difference for different routes of administration. You might have something that's starting from 1 mg per mil or 10 mg per mil, go to something like 80 mg per mil, and then go to something like 150 mg per mil, something like that. So those, you have the gamut of concentrations, and then we try to argue that, we would like to test like the low and the high, and then not have to do, stability for something in the mid range, and even that was pushed back by the FDA. So I would really caution against matrixing, because bracketing is even frowned upon. Versus matrixing is even less. You're going to get even less data from matrixing, and then if you get any one data point from matrixing that doesn't meet specs, right, then you don't have anything else. And you may have your other samples, you may collect them, but you may not test them, and then you would have to go back and test them all, kind of thing. And then if those samples are still representative of what they were when you first pulled them from stability, is questionable. So that's something that we did get pushback on, even bracketing, just for concentration's sake.
So more of the over the, let's see, the solid dosage tablets and those kind of things are the best examples for the matrixing, bracketing, those kind of things. One bit of encouragement though, Michael, I have seen people do rolling submissions, especially you mentioned the Department of Defense. If they're anxious to get this, they might be working side by side with FDA to say, hey, here's what we need. Can you give us something along these lines? Not to say that that happens all the time, but I suspect that it might. So we have seen some companies go along with that third batch on the promise that it's coming, almost like a post-approval commitment sort of thing. Okay, let's take and switch our focus for just a minute to something else, just to get across the gamut of stability things. Wanted to find out if anybody had any questions around samples and inventory. Switching over to operations for just a minute. Feel free to raise a chat question or to raise your hand. Yes, Sandy.
So yes, I'm interested in how inventory, is there inventory systems available? How is that handled? I'm fairly new to the stability world, so and I work at a manufacturing site, so I'm trying to learn what I can, and I'm curious to know about inventories, how it's handled.
Okay. And let's see, do we have anybody in our panelists wants to hop on that? If not, Walter and I are fairly experienced. Chris, you have a response?
So I work in medical device industry, and I'll put a plug in for Novatech. So we have their stability limbs, and part of that is sample management. So they do have barcode labeling that you can use. It works pretty well. So when you get your samples, you label them, put them in chamber X, so that's all noted in the software. Then when you go to do a sample pull, you just simply scan the sample out. So it's at any point in time, how many samples remain in your inventory for a given chamber, things like that. So it's we've had it for a number of years. It works really well for us. again, that's included in the, Stability LIM software.
Well, I would assume there's other manufacturers of various limbs that could include those kind of stability inventory packages as well.
Sure.
But Tamara, you manage samples and storage on a commercial level. Do you have any particular take on inventory systems?
Who is that for?
Bashar.
Oh, Bashar, sorry. I thought you were asking me. I'm like, I don't know.
We use inventory system called Freezer Pro for our biologics storage. We do incorporate it into our stability stuff as well, but it's mostly used for our biologics stuff because a lot of our biologics clients are very they require specific validated systems, and Freezer Pro gave us the validation package, as well as we were able to kind of manipulate the package for what we specifically needed for storage only, because you can also utilize it for aliquoting, or also do I don't know all the exact settings of it because our operations team really works with it. I kind of worked with the just the storage side of it when I was working on the validation of getting it approved for our specific requirements. But Freezer Pros, we've used that. We've also used another company called LabKey. The only drawback with LabKey is they don't provide you with the validated system if you buy the initial. So they have two different programs. The initial one where it's very simplistic, and then the more rigorous one. And the more rigorous one has a validation package, where the initial one has no validation that they provide us with. So we'd have to create our own validation process for that one.
Could I ask a follow-up of you guys or whoever else wants to answer that? Could you clarify if those systems link your inventory to the stability study? Meaning you start the study, then you select the inventory, it starts that particular inventory, and then the data generated to that, so it's all linked together rather than an inventory separate from your stability system. Does that make sense? Could you clarify if those systems have that or not?
Can you clarify a little bit? So are you saying if you're pulling samples in a certain timeline, can you input information into Freezer Pro or LabKey?
Yeah, generally when you create a study, I'm using terms from my old days. You're documenting your conditions, your protocols, everything in the system, and then you start it. Is there a link between starting that study and your inventory system so that the two are connected? Or are you starting the study in one system while your inventory is managed kind of separately?
So anytime we get an inventory list from our clients, we input that Excel spreadsheet, for example, into either LabKey or Freezer Pro. And then every time we start doing polls or any kind of things like that, all those polls that get pulled from the actual box will also be linked to that software box, the digital box. So if you're pulling 50 samples today, those 50 samples are also pulled out of the inventory of the software. Then if you're adding new samples into it, you're also inputting those new samples into the inventory and showing where it is in that freezer or that chamber. Does that help?
But I think for me, the question is that doesn't drive the testing time points. That system with your inventories, it doesn't drive to say, hey, now you started the study, it's 3 months, you need to go pull 6 samples. That's a separate system.
We typically just have our clients request those samples, so we never actually have to. We never know when they need to be pulled. So they give us a requirement. They'll say, please store this for 1 year, and within that 1 year, we will request X number of samples, throughout various points of that year. So most of the time, our clients provide us with the pull times and pull requests.
Okay.
Jump in with some responses that are coming over chat. Tina Dean tells us that they use their LIMS system to manage the stability inventory as well, and that their system does link the inventory with the study. And Jacob responded he's familiar with a Trackit, TRACK, capital I, capital T for stability inventory, if we're keeping track of, no pun intended, the names of these systems.
Yeah. No,
So-
Novatech Stability LIMS does that as well. So it's an integrated, I think Walter was asking, is an integrated, you know, system or two separate standalones. But yeah, it's integrated into the LIMS.
Yeah, I have an upcoming presentation on the artificial intelligence impacting stability, and one of the things that we've been seeking over the long period and hasn't been available, but hopefully it comes through with the AI packages, is that on any given excursion where something goes out of specifications temperature-wise for a while, the computers grab that, track it, apply it to your study, put in footnotes in your studies that this particular chamber was out for so many hours or days or whatever, and links it to the report that says what the impact of that might be.
Wow.
So it'd be neat if it did that for everything that's in your chamber, because
Yeah.
you might have some things that experienced it on day 3 of your calendar, and then a whole lot of new studies came in on day 4. They're not impacted, but makes you wonder what was in the chamber at the moment that you had a problem. So lots of possibilities, and I think lots of good things coming down the pike.
Yeah, I brought that up just because I've seen well, 3 different examples in my previous company where we had to customize the stability system in order to do that. It had an inventory system, but it didn't link. So just something to be careful of when you're shopping, to make sure that they're truly linked. And like John was saying, for reporting, there's a ton of value so that you can send a report of all your studies and exactly how much is in their chamber, for example, and when the next intervals are. So yeah.
Mm-hmm. We have a comment from Jacob that the Trackit system is not integrated, but they tied it into their GLIMS using the study number. So you can make those kind of connections on your own with some clever IT work.
Mm-hmm.
That would work. Just to remind people that you can raise a new topic by a chat or by raising your hand. If not, I'll continue to kind of move from area to area. Sandy.
Well, I have to jump off. I have a meeting at 10:30, but I would like to come back on at 11 because I do have another question. So
We'll
if
be
you're
here.
still on, you'll be here at 11. Okay.
Yep.
I'll join you all back at 11.
Okay, thank you. Any last things for Amy? She has to jump off as well. Was there any particular guidances that speak to cell and gene therapy? I know the biologics has get addressed a lot, but I didn't know if anything's come out. I was just in another meeting where they were saying there's nothing. So I was just curious, is that true that there's nothing addressing cell and gene therapy just yet? Amy, you're still muted as well.
So I think that there are guidances for cell and gene therapy. Let me see if I have anything right off the bat. Yeah, there's CMC information for new therapies, new INDs. Those are the guidances for industry. You can check on FDA. Check on the FDA website. There's lots of guidances.
Yeah. We try to capture those in Stability Hub as well, but
Yeah.
yeah, they're coming out all the time.
Yeah, there's lots. I mean, a lot of them are maybe around 5 years old, something like that. But they still apply. And they do tell you a lot, you know, what's required for the IND, and then subsequently, you know, what when you're going to be filing the NDA, and maybe a little bit less on that.
Okay.
I can say my experience, because I've worked in this field now pretty exclusively last 7 years.
Mhm.
It started out being as a not strictly inspectable because it's a little bit of an oxymoron. Cell therapy products are made 2 bags per patient. One is the treatment bag, and the other is the reserve held at the manufacturer in case there's a break during shipping. It might be that there's 2 containers to get enough of a dose. In rare cases, there's 3 containers to get enough of a dose.
Mhm.
And it's short shelf life product. So there are caveats in the USP as well as FDA guidances for not just rapid bioburden testing, but also the expectations for stability for a short shelf life product, especially where the batch number is essentially 1. The initial take of CBER was they were not looking for this during BLAs nor pre-license inspections. But about 2-3 years ago, they did start asking routinely, and manufacturers have been asked and given feedback that their BLA was incomplete because it lacked stability information. So this still remains a bit of a hot topic because the expected shelf life of a cell therapy for an autologous product is really only 45 to 90 days. After manufacturing, it's generally infused within 2 to 3 weeks, and only the backup sample, should you lose it during shipping, need to be used. So that might add another month to the lifetime of the product. So, however, the other aspect to it is the science aspect, which these are held in the dry phase of liquid nitrogen. And so the amount of the potential product degradation is minimal at best. One's more concerned with container integrity during handling and shipment. So the focus has been more on not necessarily the regular, and you really can't do an accelerated study on a cell therapy product. It's been more on temperature excursion support and focus on shipping validation, particularly around container integrity.
That's great input. Thank you. I think we're going to tap you for our panelists next time as well.
I 2 products. The first we submitted without, CBER didn't even comment. The second, when we did, it was, quote, missing. And, the discussion was, yeah, well, so what?
Uh-huh. Okay,
So
well, thank you.
that will all change, though. In fairness to the agencies, this will change when we eventually have universal cell lines and we are able to manufacture allogenic cell therapies. And then we'll move into how the guidances currently address the stem cell and cord bloods parts of the, as we say, ATMP industry.
Yep, I think our horizons are expanding rapidly here. Just looking at the chat comments and stuff, we have one from Jacob we're going to come back to in just a moment because Tina has raised a biologic related question, so I think we'll hold on to that for a minute. Question around biologic finished product stability using aged API, which most of the stability folks are familiar with that. How many batches are needed, 1 or 3? My initial response would be, I think we've only done 1 along those lines, and I've seen some preference for more than 1. So anybody want to comment on that? 1 or 3 batches using the aged API. So the concept there is you take an old API, put it in your batch, and really stretch that particular API because you're now using it for the shelf life of the entire product, not just the API. Anybody want to respond to that?
So I guess I would ask for clarification, that's an end-on-end study, correct?
Yes.
Yes.
You categorize it.
Oh, okay. Yeah, no, I like you said, John, I've seen 1 or 2. I've never seen 3 that I can recall.
Yeah. Well, the Russian authorities are asking for 3, along with China. And so we are going back and forth. Yeah.
Yeah, Russia always asks for everything.
Yeah. So we're just trying to justify just 1, and I think we can, since the ICH is moving really heavily into risk-based prior knowledge and scientific justification for our stabilities program from now on. So, but we're still waffling back and forth, right? 1 or 3. And in this fast-paced world where things are not in development very long, so we don't have a lot of development stability, and so we move things over to the commercial side. It's almost virtually impossible to get 3 batches on some of our products because we're already having difficulty, you know, manufacturing enough, right? So I actually have a meeting after this, and the whole topic is, do we need 1 or do we need 3? So I'm pushing for 1, so that's why I asked that.
Yeah, I had a follow-on kind of question related to that, because that's not something I've seen done before at where I work. They did for a new small molecule product, they put the API at accelerated conditions to kind of, like age it, if you will. And then now we're doing an end-to-end study, putting the drug product on stability. I've not seen that done before. I've never heard of it. So I'm just curious if anyone else has kind of, again, kind of related question.
Maybe we should have Walter chime in on that.
Well, being mostly on small molecules, no, I haven't.
Okay, me neither.
But my first thought was, sheesh, it should only be 1. It would, from my old days, it would have been basically almost a voluntary R&D type study just for additional information. But sheesh, if China and Russia are saying they want 3, there's not much you can do.
I'll bet we can now predict North Korea and Iran as well. Okay, so not to miss Roland's follow-up question was about actually subjecting the API to accelerated conditions to get it to the edge, apparently. I've not heard of that. I would think that's pretty dangerous a practice to do, but maybe I've not been following up on how that works. Maria.
I don't think this is exactly what you're asking, but I have heard of people doing that before in use stability studies. So basically trying to age it so it's close to the end of shelf life and then doing in use stability. Different. I know it's a different use, but that's what I've
Yeah.
heard of.
Yeah, because no, like was previously mentioned, like there's always a race to, get a product, to the agencies for review and approval, and you don't have the time to age the batches. So and then in studies, at least from my experience, it's usually done later on if you can get away with that. And I just learned, like, well, this is a product we kind of really raced it towards to submission, and like, well, how did you have an aged batch when we just have, like, the PPQ batches at they're only a few months, 3 months or 6 months. And so that's what the solution was. And I'd never seen that done before. So I'm kind of curious if anyone else has seen it, and if that's, something that's been submitted and been approved. So
we've done something similar. Again, I work in devices, so it's a little bit different world, but like hold time studies of some of our, what we call WIP, so it's components and subcomponents and assemblies that go into the finished good. So we've actually accelerated age, that WIP, and then we convert it to the finished good, and then we do real-time aging of the finished good. Again, it's a bit different than small molecule, but we've done that and have had no issues with agencies.
It looks like we're seeing a growing trend then, which is typical of PSDG, to hear the stories of what you think is not possible suddenly is becoming possible and more acceptable by various agencies. So taking note of this, I guess I wouldn't rule it out that it seems to be a potential trend. Let's go back and grab Jacob's question before it gets to be too far up the chat list. What is the best way to statistically analyze comparability when changing formulation, for instance, when you have very limited data? I know exactly who could answer this question, and she's in another meeting right now, so that's not going to happen. Anybody want to tackle that one? Best way to statistically analyze comparability? So we have our statisticians that usually participate with us today all were in other meetings at this moment. So I'm not sure we have a really good answer to your question, Jacob.
Totally fine, worth a shot.
Yeah, yes, sometimes it comes out that way. There is an answer. I know that 2 or 3 of these folks are going to be at the Kennex meeting in Philadelphia in June. I don't know if you have any plans for that, but there'll be a whole bunch of statisticians there. And the last couple of years, we've also had statisticians focused meetings in November that Kennex helped sponsor. And it's been 2 or 3 years running now, so I don't know if they'll change topics this year or they'll go with statistics again, but probably by then you need the information. So I'm not sure what to tell you unless somebody else pops up with an answer for that.
That is no problem. We're working with our own statistician too. I just thought I'd bring it to the wider group and see if anyone has any experience with this.
Yeah. The other thing we can do is make a note of your question and post it on Stability Hub, detached from your name and organization.
Mhm.
See if anybody that peruses that forum has a response for you.
Yeah, that'd be great.
So we'll make a note of that. Roland, general time frame. Question regarding general time frame, industry norm, expectation for placing small molecule products on stability after date of manufacture. That is a perennial hot question.
Yeah, I could give you some more background. Actually, I asked it on the forum, and you were the only one to respond to John. So I thought I would kind of show up here and ask it if other people had experience and just give some background. So I haven't heard people mention where they're working. Is that okay to do or no? Or we're not sharing?
It's not forbidden.
Oh, well, that's fine. I mean, I don't need to mention the company
For
per se, but
PSTP purposes, we try to not capture in the record, the permanent record, where things came from, and we basically have a disclaimer that people sign when they come to a live meeting that they
yeah.
won't get any information back with a name or a company attached to it. So we're
Okay.
swearing
No, that's fine.
not to pass that on, but yes, it's possible. It's a
Okay.
little bit risky.
No, that's fine. I'll leave the company name out. That's fine. This not so I have a varied background in small and large molecules and worked for several companies most of my whole career, pretty much 25 years. And the company I'm at now is so we have a challenge with getting products and stability in a timely manner, meaning it takes us around 12 months. So to break that down a little bit, it takes a couple months for product to be released. And that can vary, but on average, I've done the numbers collated that. So that's about 2 months, and then the bigger chunk of time, the biggest chunk is packaging. So we have multiple commercial products, and we only have 2 packages, and there's competing priorities. We're trying to avoid stock out. So anyway, stuff just doesn't get packaged in a timely manner. On average takes about 8 months or so. So right there you have 10 months. And then additionally, once material is packaged, then it goes to the stability testing site, and that can be another several weeks to a month before they actually station it and get in the chambers. So we've not had an issue, but there was an audit last year, I think early last year, about a year ago now, I guess. And the FDA made a comment about, oh, you guys are taking too long to get stuff on stability, and conversation went elsewhere and didn't get back to it and didn't get an observation or anything like that. But I've been trying to express to management, like, this is too long. It's been recognized that we take too long, but there's been no urgency or priority to change our practices to make it happen. And so I was a request from senior manager, like, well, to benchmark, like, what are other companies, people doing in other companies, and what is their practice? Like, how long are they taking? Again, small molecules, not biologics. Biologics usually pretty quick. So I'm just asking that question, because it'd be nice to come back and say, hey, well, other companies are taking whatever, 3 months, 6 months, whatever the number is. If we can, or a range, would be nice. People can give some input on that.
Sure. Let's see, Walter, you want to try doing your magic on a chat poll? The other alternative is for those of you who are working for a pharmaceutical company or a biologic company or something to just unmute and tell us your experience with how many months. I know a lot of people in the FDA would love to see all of us get it on within 30 days. But as Roland said, sometimes you get things shipped halfway around the world, you get a sterilization happening someplace else, another packaging step goes in. I would think that you could probably justify 6 months if you had to with moving things around. I haven't heard 12 months so far, but I can understand why it takes so long. But yeah, I would put an effort in to try and bring it to 6 and have a justification for that. I've seen a lot of people give me feedback and say, no, that's ridiculous. You should always have it within 30 days. And I'm thinking those people don't do it on a global level. They're all in one plant on one campus. And yeah, you can figure out why 30 days can happen, but
Yeah.
real world
Yeah.
situation is different.
Yeah, John, we use 6 months. And yeah, it's just, and most of our products, it's not an issue. It's within 30 days, but for our small molecule solid dosage forms, they get manufactured in one country, packaged in another country, and then sent back to a third country for stability. So it can take 6 months. And that's what it is in our global standard, that you have 6 months to start stability.
Okay, and you have some justification documents here and there that's lined up to show the inspectors when they come through?
Yeah, but we've never been questioned on it. Honestly, we've never been questioned on it. But I mean, we've got plenty of data because, again, our stability start time doesn't, I mean, our stability studies don't start until they're in the chamber. So we have plenty of data past expiration. So we're showing that the delay is not hindering the product in any way.
Yeah, that's a good point, Tina. Yeah, so the products I'm working with, they're legacy products. There's 4 specific products in particular. The oldest one's like 10 years. Been a commercial product for 10 years. The newest one's about 2.5 years. So we have a lot of data. The products are very similar, but there's minor changes in the chemistry from one product to the next. But they're all kind of built foundationally in one, and then they just created others and mixed them together. Yeah, no, that's just and that the form is tablets and granules. I forgot to mention the form in blisters and sachets. Go ahead. Sorry.
I was going to say, let's try a poll for now. Sorry.
Sorry, John, can I come in? And this is Jeff, but Thermo Fisher Patheon, we're manufacturing mostly tablets, capsules, oral dosage forms. I would say typical times for getting into stability chambers would be about 2 months. Where it does impact us is that sometimes packaging does take longer than that. And according to our SOPs, the way it works is that when we put something on stability, where we use the initial release results as our time 0 stability data, provided that we can get the samples into the chamber in no more than 1 month after doing the testing. If it takes more than 1 month, we have to repeat the time 0 testing.
Okay, so I'm just gonna post a question, and if you would try to answer it via chat. We'd normally like to have a yes or no question where you raise your hand, but if you could just pop in the chat, and we'll just take note of the responses. So if this question applies to you or you are able to answer it, would you please just reply the number, how many months are required from date of manufacture to placing the product in the chamber? We'll just give it about 30 seconds here.
While you're doing that, if you can chew gum and do the response at the same time, I'll do a quick reminder for those who have joined after the initial start. Our panelists include Michael Brommer, Sales Manager for Parameter Generation Control Chambers, Jeff Carr, Retired Director, Analytical Development, Thermo Fisher Scientific, Patheon, and contributor to Stability Hub North America for correspondence. Bashar Deeb, Quality Manager for Precision Stability Storage with facilities in California, Florida, and Massachusetts. Maria Krish, Research Fellow, Physical Sciences from Freethink Technologies, and they're the owners of ASAP Prime. Chris Latos, Manager of Stability Services at Hollister. Myself, former Director of QC Stability at Regeneron, PSGG Facilitator and Editor of Stability Hub. Walter Routh, Retired Director, Global Stability and Reference Standards for AbbVie, and also Associate Director of Stability Hub. Stephen Schwartz, Director of Business Development East at LAB Validation and Monitoring Solutions. And we saw Amy Wong, who was no longer online at Arcutis Biotherapeutics. And Christina O'Neill, former Executive Assistant from Sanofi, is helping us coordinate. So what do we see here so far? We have a couple of 3 months, 6 months, 2 months, and we don't have one. We have to repeat time zero of more than 60. So it's kind of a open door kind of thing, but the safety net is repeat testing.
Sure. Now, just to confirm what we're talking about here. So for example, if you go with 6 months, then you would end up testing your product 6 months beyond expiry. Is that correct? At least?
Yeah, we've actually had material go on stability. Like I said, it's around 12 months. It varies. I kind of track everything. I mean, I'm directly responsible, but I kind of project coordinate. But we've had one lot that the oldest one was 17 months when it went on stability. So yeah, we run all our studies out to 60 months. So all the time frames, those are study time points, per ICH 3, 6, 9, 12, and well, annual lots with the 6, 12. We had removed some of the time points, but so yeah, all and the product's stable, as as most probably know. Small molecules products can be stable up to 10 years or beyond. So yeah, we just put them on whenever it goes on, and whatever that time point is, it's we represent it as a study time point, but if you add on from data manufacture, that can vary from can vary up to many months. But we just test throughout, and most of the products we don't have any issues.
Okay. The reason I bring it up is just from a previous life. It was anathema in our company to test beyond expiry. Upper management's dictate, which put us in a really odd position compared to the rest of stability industry. Well, just to say, we had a process to explain that, and we actually never got an observation on that. Never even got questioned on how come you didn't go beyond. We're not in the chamber for the full period. Surprised, we were. I always consider that one of our highest risk situations, but it never came up. Never got. I mean, we didn't hide it. It's right there, data in chamber, and so I just wanted to say that there are ways to prevent that. The issue was simply the risk of an out of spec beyond expiry, which is a whole bunch of trouble for no reason, essentially.
Yeah, agreed. Well, that's what management's falling back on. Like, the products are so stable. When we put on stability, it doesn't matter. I mean, in general, that's kind of the thinking. Like, but then if you did have one particular lot of an issue, obviously an annual lot representative of the whole year of a particular manufacturing site or in that particular packaging configuration, whatever. And you didn't put it on till 12 months after, then you see a problem, whatever, 12 months from there, that's a big issue. But no, I appreciate the feedback here. So it looks like the range is from, well, ideally 30 days as discussed, but 2 to 6 months is what is actually people doing in practice, it seems. So
yeah, for the most part.
Yeah. Okay, great.
Let me toss it over in a different direction, since we have such a wide variety of interest from our audience, and whatever people jump onto, we'll keep on going with for just a bit. But I wanted to get into stability chambers for for a moment. People have questions on that, by all means, plop it up in chat, but I wanted to open up that discussion a bit by saying for validation and requalification purposes, we've taken polls in the past, and some companies used to requalify or revalidate every year. And then suddenly it went to every 3 years. And then we found a smattering of people every 2. There's actually a guidance someplace that says recommended every 2. And then lately, it's the continuous validation or continuous mapping that seems to be where technology is taking us. So I was wondering, Stephen, can you comment a little bit on the trends you see there? It shows that you're unmuted, but we're not hearing you.
No.
There we go. Nope, still can't hear you.
Sorry, can you hear me now?
No, we can.
Yep.
Oh, I'm gonna have to reset the mic. Can you hear me now?
Yep, we're good.
Yes.
All right. Sorry about that. It sat so idle for a while. It decided not to work anymore. Again, from the industry, I visit a lot of places, and it really depends on how conservative you are and how often you're getting audited. That's kind of what I see. Specifically, if you get an audit a lot, people want to make sure their process is in control, so they're doing it yearly. And again, that's super conservative. Generally, I think the sweet spot is actually like 2 to 3 years. And then certain industries, they push companies that are not as regulated, they'll push it out to 5. And again, it's what you feel comfortable with. And again, how often you are audited. And again, if you're doing something that's more of like a third party or storage versus the actual company itself that's manufacturing it, you'll see it actually push out a little farther. They'll push more to the 3-year mark. I've also seen people do it based on risk. Usually stability is risk priority number 1. So then you'll actually see it more frequent where they're pushing almost up to a year because they consider stability the highest risk. Again, that's what I've seen in the industry.
Yeah, question came in from Laura. I've not heard about continuous mapping. Can we explain that? A little bit about the equipment that you use for continuous mapping and why people would do it.
So the technology is going in that direction. I don't believe it's there yet. I mean, I could tell you right now, we're developing the technology to do continuous mapping. And again, a lot of the problems is the data storage. You need massive amount of data storage to handle that. So technology wasn't there in the past, but it is there now. So technically, like if someone does a requalification using 12 sensors, right, throughout their unit, and again, that's in the ISP guideline. Ideally, if there's a way to mount the probes inside your chamber permanently, you could actually do continuous mapping. And again, data has always been the problem with trying to perform that. But nowadays, technology's gotten there where you can handle massive amounts of data and sending. The problem with a lot of this, though, is if you want to go Wi-Fi with this, there's a lot of data getting sent back and forth. And then all of a sudden you have interference causing problems. So that's another issue that hasn't been solved yet. So no one wants a false alarm, right? Because with all the data packets getting sent, you'll have a bounce here and there. I'll give you a false alarm. Again, you could create an algorithm to throw that out. But again, loss of data is not a good thing. And again, in the industry, that's kind of very frowned upon. So again, that sort of technology is still getting developed, but it's right around the corner. People are about to come out with different systems that can handle continuous mapping.
Okay, well, thank you for that. Yeah, the kind of thermocouples and those kind of things that are just standalone, like you mentioned, Bluetooth. that can send the signals out. You just replace them periodically. It has a wide appeal as opposed to having a crew come in on a regular basis if you own the measuring devices yourselves. It's neat. But like you said, if you gather tons and tons of data, I kind of like the idea that you can compress the data and show a line that's going across that might even cover a year if nothing went wrong. But as soon as it showed a hiccup or something, then you would expand it down and look at some data that appeared every 5 minutes until the incident was over. I've seen something along those lines, but yeah, like you said, there's pros and cons of doing that. And we have run into some people that only do it on an as needed basis that would be equivalent to your risk management or risk assessment. If you change something significant in the chamber, then you want to remap to do that, whether it's placement or equipment, that kind of thing. Can you recommend any publications or guidances that people can look at as to how they should be dealing with validation and of chambers and placement of things? I know ISPE, I think, might be the major source, but you typically don't find like an FDA guidance about stability chambers.
No, I mean, I always recommend using the ISP guideline for controlled storage. It gets down to the details that most of the other guidances do not. And again, not stability, but other types of chambers too, or warehouses. I referenced the WHO guidances, but that wouldn't be more for stability. For stability, I go by the ISP guideline.
Okay, thank you. Anybody else have a chamber related question? If not, we'll switch the focus a bit. You can always come back to it. Let's go over to the laboratory and chemistry, just to see if we have any questions along those lines. We have Jeff standing by to field some of those questions for us. Let's see, one of the ones submitted earlier. Is it a regulatory requirement that analytical procedures used in support of an NDA for stability be fully validated, but for earlier development stability studies, how much validation does the panel recommend? Bearing in mind that the formulation product details not yet been established. So we've seen a couple of questions like this come through to StabilityHub, where it's early on and the guidances fully apply all the way through, or do we have these graces that can be applied early on? Jeff, you want to take a crack at that?
Yes, okay. Yeah, so according to the regulations, you definitely have to have all your methods fully validated by the time you get to your registration stability for filing your NDAs and beyond that. But in earlier development, a lot of stability is going on also in support of formulation development for your clinical trial materials and things like that. And you need to be doing stability there. So how much validation is required there? Now, historically, people have done some validation. They might call it qualification, where they do maybe some quick and dirty type of validations to be comfortable that their methods are okay. We do now have the ICH Q14 guideline on method development, which does tend to indicate that you maybe ought to be doing something a little bit more rigorous than that. My suggestion is that you need to consider what's fit for purpose here. Now, if you're in early development, you're supporting formulation development. Your formulation development colleagues are pretty heavily dependent on you, providing them with reliable results as to which of their formulations and their processes are producing the most suitable results. And so very big decisions are going to be made on the results that are going to come out of these early development studies. So for me, the most important thing is to know that the methods are actually stability indicating, and then I'm actually seeing the degradation products that are going to really happen later on in the product life cycle. So doing some quick and dirty forced degradation is usually a good thing to do to get some comfort that the method actually does is sensitive to the formation of degradation products. Another approach, which I highly recommend, is the use of software for this. So we have predictive software out there. It comes out of a company called LASA, which is associated with the Leeds University in the UK. And they have generated something called Zenith software, which is a chemistry expert system, and you create an organic molecule and it will predict what the degradation products are going to be under different types of stress conditions. So you can tell it you're going to heat it, you're going to hydrolyze it, you're going to oxidize it, you're going to photolyze it, whatever you're going to do, and it will predict what type of molecules are likely to happen, with also an indication of how easily that happens. So it gives you a reasonably good idea of what the sensitivities of the molecule are, and it's quick. As long as you know how to use it well, about half an hour or so, you can get your answers. And so this is a very valuable form of information to have. Then you can do some of your quick and dirty force degradations to see whether what you're getting from those results is consistent with what you're getting from the Zenith predictions. So for me, that would be the most important thing to do, although I might also want to do a bit of repeatability and accuracy determination also to get a feel that the method does work reliably.
Okay. Thank you, Jeff. You said a key buzzword there for accelerated predictive stability, and that makes me think that maybe Maria would have a few things she could add on to that, especially how it's used at different parts of the product life cycle. And got any comments about that as to how regulatory agencies view that and what's been most successful and what's the most challenging?
Well, I think the Zenith software actually does sort of a different thing, as Jeff was saying. That's more of a complete computer exercise and I completely agree. I think that's a great way to, I think we see the same thing from our perspective as the stability indicating method development is the most important. I mean, I guess, were you asking about accelerated predictive stability as a separate question or linked to this first one?
Well, his comment sparked my thought. I'm
Yeah.
thinking separate question, but it's in the same ballpark. It made me curious as to I can see where you want to identify what those products are, but if you're using accelerated predictive stability parts rather than just identifying the materials, how is that working?
Yeah, so I mean, the accelerated predictive stability are based on, they're not just a computer exercise, they're based on experimental data, if people aren't familiar with it. So it's a short-term experimental stressing. It usually gets, if you're thinking about stability indicating method development, it usually gets a much smaller sort of universe of degradants than, a forced deg experiment that's traditional or the Zenith program would predict. It's usually the most important one, so it can be used to assess, assess what is going to be important for stability. From a point of view of method development for it, you still need a stability indicating method. So I think I'd still agree with the basic, assessment of the stability indicating method is the most important first step, because again, you need to be able to see things growing. And then usually what you see in those studies are a subset of what you would see in a forced degradation study, but they're the ones that you would want to focus on. So from a regulatory perspective, these kinds of experiments are used all sorts of ways. They're used in a non-regulatory way, in early development for formulation screening or picking an API, or potentially, evaluating, kind of packaging that might go up on more traditional stability. And in a regulatory sense, it's generally used at early stages to kind of get into the clinic faster and support product understanding. And as it goes to later stages, there's a number of different, like post-approval, showing that a change is minor, or, from a perspective of like justifying excursions, those are usually within the design space, and all sorts of sort of supporting applications in various different ways.
Okay. And then I recall when the program first came online for something like ASAP, it was kind of new and regulatory agencies were just getting familiar with it. And then reports over the years are that it's gained a lot more acceptability, especially if you're doing comparability kind of things along those lines. And I'm imagining at some point that the acceptance will go further down the road.
Yeah.
I'm not sure we'll quite ever get to, you don't need to run stability unless if you're doing ASAP. But maybe someday, who knows, that artificial intelligence will finally bridge the gap for us to do that. So can you comment a little bit about the types of products that those accelerated methods are best working for and ones that they're not yet?
So the most common use, I could say, is definitely small molecule drug products. You know, chemical changes in small molecule drug products. As you get larger, what you're looking for is a method that has sort of a small RSD. So you need an analytical method that's got, you know, enough precision to really make a good model. So by far the biggest use, and again, this is largely for, we sell software, so our customers use this software and other companies. Biggest use is definitely for chemical stability for small molecules. There have been a number of successful uses in larger molecules. It really depends what stability indicating. And then there are some other physical properties that have been shown to follow similar things. So it has been used in dissolution, color change, viscosity change, actually recrystallization of amorphous material. Those are much less common. So a lot of the uses are non-regulatory. I have heard of regulatory uses for at least the dissolution and color change. But yeah. But
Okay.
the bread and butter of what people are using it for is small molecule, but any number of different forms like oral solid dose, liquids, gels. Things.
Okay. Thank you for that.
Yeah.
Yeah, John, we had some experience of using ASAP at Thermo Fisher Patheon, and exactly as Maria says, but I would just point out that for me, the most useful application of this was to support formulation development, where our formulation development colleagues will come up with a number of prototype formulations that they wanted comparative data on. And we ran these through ASAP approaches to get which is the best formulation to take forward into further development. So this was our most primary use of this approach.
Yeah, and I can really appreciate what that's done for the industry over my 50-year career ending this July. And that when I first started, you would get the people in the head office had about 10 different formulations and colors and flavors and things, and that you had to put every single one of them on stability and watch at 40-75 for a month or 50 degrees for a month before you could start weeding those out. So this is great technology that we have at our fingertips.
Yeah, and I agree with that, Jeff. I think that's a great point. I think a lot of our larger software users, from what I've heard, often implement it as part of the kind of early development process for formulation screen.
Okay, and if anyone has any more questions along that line, throw it into chat. And for the moment, I'm going to go back to a question for Roland. For new products transitioning from clinical to commercial, do organizations typically have commercial specs finalized and in place at initiation of formal stability studies prior to the initial submissions? So I can have people comment on that. My experience is that yes, you try to get those as far down the road, but you're always trying to tweak and tighten specifications, especially if the FDA nudges you in that direction. But in general, I believe that's the case. So maybe some of you chemists would like to talk about that.
Yeah, let me just give a little more background, John. And just the other panels. So yeah, again, as I mentioned previously, you know, most companies, you know, they're rushing to get their products submitted and hopefully approved. So you know, there's usually not much time in the schedule to allow. So what happens? Again, where I work, small molecule, the formal stability study, registration, and in a lot of instances, the validation well, validation studies have started obviously prior to submission because you need that data for the submission. But they go back, the registration studies in particular, being the lead studies for, determining shelf life, it varies by product and how fast it progresses, but in general, around a year before the submission. With the clinical specs, there was initiated with what they have for clinical specs, and obviously over that time frame, you gain more knowledge about the product and you tweak your specs, as you said, and then. But what happens is, in where I work, is like they're tweaking specs like 2 to 3 weeks before the submission, sometimes even like right before, like a week before, days before the actual submission is filed. And it's not something I've ever seen. Typically, I've seen in my experience, specs are locked down at least 3 to 6 months before the submission. So what is happening from my situation, it causes a issue with those studies and in our slim limb system, and trying to accommodate the clinical spec and the commercial spec, et cetera. So hopefully just give a little more background on that and just get some input.
Comments?
Yeah, I'm muted. Yeah, in my experience, when you get into the registration stages, you've already probably done quite a few stability studies already in support of clinical stages. So you have a pretty good idea what the product is likely to be. But of course, the NDA you're going to file with 12 months of real-time data. You have your 6 months of accelerated. You're allowed to make some predictions based on that. You might be able to double your shelf life double your real time for the shelf life at the time of submission. But then you continue to collect your data as you go on. And as you collect it, you can then put in for variations for shelf life extensions. And in my experience, that's probably the way that we would have gone about doing it.
Yeah, agreed. But I guess it just to like the specific question is like, what time frame? Well, are the specs like pretty much finalized before those registration studies start? Or again, as John mentioned, and I've seen, we do it regularly, we tweak them as we got more information that's maybe justified to change it along the way. But are they pretty much locked down? Because that's not what we do here. We just basically defer to the clinical specs until we get to just before submission, and then they take another look at it and like, okay, well, maybe we can we can widen this spec or tighten it or whatever the case might be.
I'm holding back to see if anybody else.
Yeah, from the requirements, I think it's in Q1E, isn't it? In reporting stability data, you're allowed to have double the shelf life with 12 months of data, provided your accelerated data has all been solid. They say that there's little or no variance, whatever that's supposed to mean in the data. You can then go in with a 24-month shelf life at the initial time of registration. Mm-hmm. They may or may not accept it, but they're not going to give you any more than 24 months. So it is pretty well locked down at that time. But then, as I say, as you move on and get more, you continue the stability study. As you get more data over 2, 3, 4, maybe 5 years, you just extend the shelf life. I thought that's the way it would work.
Yeah. Overall, I think it's a pretty safe bet that they are locked down to some extent at the time period where you're referring to. I worked for a company that had a horrendously complex product, and only 1 or 2 guys in the entire place could make it work for a method. And so they were on duty all the time because they had to do such ridiculous things or hold a vial a certain way or heat it to a certain temperature that was more finicky than official. And their biggest nightmare was once they put that in and had the method approved, they were totally reluctant to change the method to something much simpler because it had been approved. So they didn't want to take a chance on getting delayed or opening up the books again. And it was kind of self-defeating that they got to that point without having changed the method to something better. But they went with the expedient one that got them on the market and approved.
Mhm.
So I don't know which is best, to be on the market or to be scientifically better when it comes to your method. All right, so enough about that. Let's talk a little bit about audits and regulatory inspections for those of you who might have an interest in that. I have some ones that have been previously submitted, but I'm more interested in seeing if any of you are interested in finding out more about stability audits, trending of those things. Some of the notes I have is about that auditors seem to come better prepared for asking questions about stability these days than they ever have in the past. Whether or not that came from information sharing between agencies and things like PICS where they share information and techniques back and forth. Has anybody noticed that in their audits? I know they're particularly picky on inventories these days. And over the course of the years, that caused me to change my outlook on how I did inventories.
I can add
Yeah,
something.
John, we had that. Oh, sorry.
Oh no, go ahead.
Go ahead, Bashar.
Oh, okay. I was going to just make a suggestion. One thing that I've noticed from my audits from both the biorepository and the stability side is I've been using a SharePoint link where I provide the auditors like maybe like 2 weeks or 3 weeks prior to the audit with the ability to review our SOPs and like, you know, give them access to our registrations or certifications. And it seems like a lot of the times, like after they come onto the audit, a lot of the times they're a lot more less aggressive because I was like so open to giving them the ability to view those that SharePoint link. The nice thing about SharePoint is you're able to block them from being able to like download documents, they can only view the documents and not edit it or remove or delete stuff. So it's very nice to kind of give them that initial audit request. And then, you know, once they come on site or if they're doing it virtually, they're, you know, they're a little bit more less aggressive, I feel like, from our perspective from, you know, being audited in the last like 3-4 years with precision.
Yeah, it's a tremendous shift in philosophy over the years. The classical training was only say yes or no, don't answer any questions, don't give away anything, versus they act a lot more cordial if you're transparent and cooperative than trying to stonewall. Jeff, go ahead with your comment.
Yeah, yes, we certainly at Thermo Fisher were finding that FDA and Health Canada audits were getting more thorough, shall we say. They certainly knew their way around. They knew their stuff very well. Mostly they were very interested in HPLC software systems. So in power systems, they are very strict on us having full accountability of just about every injection that has been put into the system. So inspection of audit trails, going through them. If they find something where an injection has been rejected for some reason, they want to see deviation reports to account for it, and they've been going through those very thoroughly. Going back to you more specifically, what your question was about sample management, few years ago, we did have an FDA inspection. Now, for us, in our protocols, we specify how many samples are going to go into each chamber. We put in sufficient for doing the testing, but we also put in some spares because things can go wrong. You can drop a sample. You might want to do some other work on it, on some samples that have been in a chamber. So putting some spares into a chamber is a general practice, and so you have more in there than you really need. But you have to account for all of those samples. If some samples have disappeared, it's going to throw up suspicions that there's been some fraudulent practices, and you didn't like the results you got, so you did it again with another sample. That's what their suspicion is going to be. So a few years ago, we had this audit. They went through the registration stability protocol. It specified how many samples went into each of the conditions. We were doing real-time accelerated intermediate, and I think there might have also been a zone 4 territory condition real-time as well. So this guy wanted to visit all of the chambers. We had to show him all of the samples that were in the chamber. We had to show him our records for the samples that had been removed. At the time of the inspection, we were probably about 18 months to 2 years into the study. So a lot of the samples had already been removed and tested. So he was now looking for how many samples were remaining in there. We had to count them all out in each of the chambers. He then went to the protocols, and according to the protocols, how many should there have been? And it turned out that the right number was there, so everything was all right. If the right number had not been there, he would have been looking for deviation reports to account for it. But he definitely did that, and as it turned out, it worked out all right for us, but they do check these things.
Well, thank you for that. Yeah, I have similar experiences, only it turned out not so well in that they asked us for the number of samples in a bin, and we were wrong for 3 times in a row. And that led to the discovery that we had managers signing off for people to be able to withdraw samples, but they never followed up in correcting and updating the LIMS tracking system. So we're kind of going at it and doing ourselves in from lack of follow through with training people and them taking responsibility, especially at 3 o'clock in the morning. If they're on the phone and they authorize the extraction of more samples, they may not even remember it in the morning that they came in to do that. So yes, that's those are observations with audits for sure. Laura Bello says that they had the same finding, Jeff, where we weren't counting our inventory at each pull point. So there's different places you can go. I think I wrote an article a ways back on StabilityHub about inventory methods and some pros and cons of each one, and the one that I recommended the most. Let me turn to Mike.
I have a question about inventory.
Sure.
Do you do an annual inventory then, or reconciliation, or is it just during inspections that you do that? Or does that make sense what I'm asking?
It does, and I think we'll be all over the lot for there. This would lend itself nicely to a poll if we could do the poll thing, but some people use different methods. So some do an annual inventory and go through everything. Other ones might try a rolling inventory where every month they try a different stability chamber. The problem is if you're moving samples in and out during the period of your inventory, it complicates things. So we've seen different ones work. Sometimes you have your summer intern come through an inventory. I like to do one that gets updated every time that you pull a product out and submit samples. Whereas you go through, and when something's due, you count the total that's in there, the total you took out, and then do a tamper evidence seal on bigger bins where you know there's 100 in there, so you don't touch that bin until you have to. So when you do an inventory, it's quite easy to count like 5 bins of 100. You got 500 there, and 1 bin where you count 33 more samples for a total of 533. And then you have somebody sign off every time you move your sample. And then by the end of 1 year, you'll have ideally submitted at least one submission from each stability study, and you'll have 100% inventory there. It just gets updated at every pull point. So I did that once in response to finding out that we had wrong counts through limbs all the time. And a lot of the tracking systems are great at double checking how many samples you're supposed to have versus what you still have in theory. Did anybody else want to give a more detailed answer as to what they're doing with inventory that's worked really well with them?
We have a secondary person, so anytime chamber samples go in a chamber or they're removed, we have two technicians, so that they can verify exactly where the samples were placed and how many samples were removed. They're wanded out through the barcode, all that kind of stuff. So we always have a secondary person as well.
Any other comments on inventories? I'll make mention that when we had a discovery that our inventory was off, it initiated a CAPA. And of course, part of a CAPA is to look and see how widespread your problem might be. And it was applied to areas outside stability all across the site. And it turned out that 6 or 7 systems were having similar problems. And our problem was our CAPA system, whoever initiated the CAPA inherited everything that had to do with that CAPA. So a very junior staff person that was going to be responsible for the stability discrepancies suddenly was responsible for all the discrepancies across the site. And it was a nightmare for that poor person that had to do that. So we decided that, at least philosophically, that I would like to keep it local within stability rather than take a CAPA that you have responsibility for and apply it to other divisions. Any other inventory comments?
I was just curious from the contract storage perspective, especially Bashar, if there's anyone else. How do you handle your stability? The kind of the question that Sandy was bringing up. Then, how often do you double check or reconcile your inventory?
So we do a monthly QC check on our freezers. Not all of them. So we'll just pick like 2 or 3 freezers and 2 or 3 chambers like throughout the year. And each time we'll do a quick QC check. But also, anytime material is put into our inventory system, we always have to have the initiator that put all the material in approve it, and then also we have a person that double checks it, and then finally at the end QC does a final review of all the sorry, not QC, QA does a final review of everything that has been inputted into our inventory system.
Okay. I was curious then, how do you do your customers specify how often they require that there be a reconciliation, or is that you're just...
We've never had that asked, which
Okay.
is so weird to us. Even during audits, they really barely ever like ask us like, can we see your like QC checks? We always provide it to them because we just like show them like how we make sure things are done, but it's never been an audit question.
Okay.
Anytime we've had our clients come in to visit. It's kind of weird.
Thank you.
You're welcome.
Thank you for that input. For Mike Brommer, I have a question regarding stability chambers and advice to people that need to purchase a chamber. What are the common things that go wrong when somebody wants to buy a chamber? What kind of information do you like to see? Do you like to interview them for their needs? That kind of stuff. And I'm thinking back to a person who participates a lot here in PSDG. He's not able to join us today, but he tells a story when he was new and he was the chamber guy for the company. They said they needed a refrigerated chamber. And so he went out and he bought a refrigerated unit that ended up serving in the cafeteria for keeping people's lunches cold because nobody gave him any specifications. Can you give us some advice as to what somebody seeking a new chamber should be doing?
Yeah, sure. So I think some of the most important things are about the facility it's going into. Clearance issues are a big thing. We obviously most of our units we offer in 2 variants, a vertical or horizontal, and it really comes down to customer preference. The horizontal is mounted on the roof, where typically we need maybe 4 to 5 feet of clearance above the top of the chamber. The verticals are really popular because of maintenance needs. They're directly bolted up to, a front or side wall of the chamber. So you don't need to get any, if it's an issue as far as getting up on top of a chamber for safety on a facility, you don't need to deal with that. So that's an easy thing. But yeah, obviously getting a good idea on the actual expected use of a chamber is very important. Obviously, now we're talking about just storage, but another big thing that can be tough to get is heat loads. A lot of our equipment can only handle a certain heat load, and you really get up to that limit pretty quickly if you have people working inside, or if you have equipment. If we're doing some sort of equipment testing inside, you can get up to that heat load relatively quickly. So that's all really good information. Obviously, our smaller units, power isn't often a issue. If we talk about our bigger units, some of them could be 460 volt, where a lot of sites just aren't prepared for that. But a lot of the time it does require a bit of back and forth until we get all the information. And sometimes that's because the person reaching out doesn't have all the information or wouldn't know certain pieces of information that we're going to need as far as what the install logistics are going to look like, what the maintenance, requirements are going to be. So there's a lot of back and forth that needs to take place before we can accurately quote the proper matching equipment.
Or even the width of the door that you have to roll your equipment through in order to get to the room. Could
Yep,
be a
exactly.
problem.
Yeah, no, we typically offer sort of our standard is 36-inch wide to accommodate those carts, but we do see some people who want to have the option to have a floorless room and drive a forklift in and unload product. So obviously that all affects things, and obviously the ambient temperature can be a huge thing, especially dealing in certain countries, in South American countries, where an ambient temperature might get up to 40 C plus. Knowing that, or knowing if it's going to be in a conditioned space or not, those are huge things to look out for as far as making sure that the unit's going to be able to maintain condition.
Okay. Anybody have any questions along the lines of chambers, chamber purchases, chamber maintenance? If not, I can ask another question in a different area. We can always come back if I see one pop up here. I wanted to not miss the opportunity to talk a little bit about in-use tests. That's been a hot topic across the industry. And a couple of guidances here and there. We were talking about the in-use guidances a little bit earlier. I forget which group I was with to do that, but there are some general guidances, or some that are very specific about withdrawing samples from an injectable. Some are veterinary guidances. So I was curious if anybody has their favorite guidance they like to go to, whether that's a WHO document or something else, and the kind of application they have. And one of the things that I've been wrestling with is if you have like a combination product, such as an infusion pump, it gets used once. It's in the clinic setting or a doctor's office, and maybe at the beginning of the day, in anticipation of a patient coming in, somebody might unwrap the product that has a cartridge that plugs into the pump. The pump might be attached to the body for a number of hours. I mean, maybe the person gets set up but doesn't get started. So there's a number of conditions of light and body temperature and that kind of thing that this can get exposed to. And I'm thinking this is a good time to run through a dry run where you don't actually use the product, but it gets exposed to light and temperature and that kind of thing, and maybe some hoses and a cartridge or two. It gets recollected and then sampled and tested. Now, that is not described in a use test, but I'm thinking the way that regulators are these days, they might actually ask a question or two about that. So I wanted to see what are the kind of applications you see for use tests, and is there anything that brings clarity to your use of those tests, or any confusion you see coming out of guidances? Seeing a lot of puzzled faces. I can be more specific. Yeah, Jeff.
Yeah. Like I said before, we are doing solid oral dosage forms, and some of the examples we do are powders for suspension. So you have a bottle with a powder in it, which is manufactured, shipped off to wherever it's shipped off to. And when it's dispensed, what's going to happen is it's going to be reconstituted, probably with water, and it's going to be given to the patient to take home, and they're supposed to take a dose out of that bottle. The bottle will contain, say, 1 month's supply of the suspension product. So the in-use study we do would be to do a reconstitution of the bottle and then put it aside, and then every day we would shake it up. We take a sample from it, and what we're looking at is suspendability and consistency of dosage. So if you have to take out a volume and show that it contains the right amount throughout the month. We also would do a related substances test on the sample, and we do dissolution testing on the sample. And the dissolution one has its own problems in it, because introducing a suspension product into a dissolution vessel is a little bit of a problem. But what we do is we introduce it under the paddle so that it doesn't just disperse into the volume. And so that's an example of a in-use test that we would do. Don't know of any guidance out there that actually describes that sort of thing. I believe there's EMA guidances which tell you have to do in-use testing, but it doesn't go as far as specifying just how you do it, to my knowledge.
Yeah, as I recall, it mentions trying to do some of those towards the end of the shelf life or end of a batch study. To do, I guess it's something that it varies from guidance to guidance as to when you do it. But I find it interesting that it's getting more and more questions from stabilitarians around the scope of industry. There was one specific one from one of our subscribers to StabilityHub, where they were worried about a small clause at the bottom of the guidance. It took me a while to find it. And that's that they had done some micro testing. I think it was bioburden especially. And it spoke about if you go beyond the recommended use time or something like that, you had to repeat the micro test. Walter, if you remember this more than I do, you might comment. But I was really at a loss as to how to respond to her about doing that testing over again. So I think maybe somebody that's specializes in microbiology might have run into this more, but I've not heard about redoing a use test. But maybe the regulators were concerned that the actual time that the product was out there available was longer than the protocol test that they had. So they went around and had to reprove that the micro was and the preservation was still good. I don't know if anybody's ever run into something like that. Or anything in general on use testing, if you're a stability practitioner and you're concerned about that. Yeah, let's see the wording of that. I think I have it specifically now that I look down here and see my notes. Does anyone know how to do a repeat challenge for multi-dose injectable products for claiming in-use period of more than 28 days? And they cite the EMA guides. That's where the little thing at the bottom was. Doesn't state on how to execute the repeat challenge. And then they gave a link to the guidance. So that's new to me. I haven't run into that before, and we'll keep on watching to see if anybody comes up with an answer in the forum to do that. Chris Latos, anything on combination products that we might be checking out in our time together here? I know one of the issues was you're putting things together in a package where one has an expiry that comes up much sooner than the other. One of the questions had to do with a syringe that expires 12 months after the drug product in it. Can we proceed to test the syringe through its expiry, but no longer test the drug product for the additional 12 months? Seems like it gets pretty complicated.
Yeah, I mean, the one with the shorter, expiry is gonna dictate, for the product itself, what the shelf life is gonna be. But yeah, I don't know why they would continue testing the syringe beyond, if the drug product is the limiting factor, I'm not sure why they would continue to test the syringe, past the expiry date of the drug product, but yeah, I'm not sure.
Okay. Yeah, it seems it's fairly complex and case by case, and a lot depends on how well people made out with their reviewers and regulators as to the impact of that. We have a comment from Mike Skidmore. As far as standalone statistics programs, Minitab, Jump, Prism, very popular, and ICH and FDA guidance says what regression charts and data analysis are expected for an FDA or a BLA. And since it's fairly repetitive and standardized, there are publicly available macros or templates for popular stats programs for the startup company to do this or on their own. Let's see, I was transitioning there from what looked like a statement to a question. Michael, you want to verbalize that a bit for us? Are you asking what programs there are? Or are you recommending some
Yeah, so
ones?
typical small virtual company question. Money's tight, and there's certainly stats programs that the analytical development team is already using, but since they hadn't yet purchased a complete stability program and can't take advantage of that, we got into a discussion about, well, since these things are fairly repetitive, can't we just use one of the existing stat programs we have? We had Prism in our case. And I was wondering if some of these popular statistics packages on some of the public forums, there's available templates or macros to just go ahead and do this expected data analysis, as opposed to setting it up from the beginning every single new client or small company I get involved with.
I think a lot of companies are using these packages, especially Minitab and Jump. I see that a lot. And it's common, but there also are commercial packages, StabStat. And
Yeah, so in our case, the drug substance manufacturer, a CMO, used a commercial package, but only for things they did. And then they said to us, well, for the long-term shelf life and ongoing NDA maintenance, we expect that you guys will perform your own analyses to submit because that's out of scope for us. And then our packager labeler didn't provide statistics services, so we had to do it internally. So we were looking, how can we use either Prism or Jump or Minitab without buying yet another software package to just, because cash was a concern.
Yeah, I know. What you're saying is something done, like John's saying, everybody does their own. So I'm not familiar with them online. I just Googled it as you were talking, and maybe things came up, but I haven't been able to check yet. The reason I bring up those
I tried myself to do this once, and it's like every time you go to a new company, it's like, why am I reinventing the wheel? I'm sure some nice stabilitarian posted a template for Minitab with all the analyses already set up.
If any software might have that, but if there is anything for software, I would guess for SaaS, because it's so customized as it is for every single use. And I've also seen one of our, well, actually it was a customized program that was put together for us, what was that, 15 years ago, but that person moved on and I saw them making other stuff. So I don't know. I don't know the answer, but I would guess if anything, it would be out there for SaaS.
Okay.
You might try a users forum, like for Jump, there's got to be some like user groups online and just, post that question, for Jump or Minitab or whatever.
I did in the Jump and Minitab user forums, and I didn't really get any answers. Minitab has within it a shelf life estimation macro, and that was where the idea came from. But this particular project, analytical research and development team already had several Prism licenses, so I was trying to not buy yet another package for a company of 12 people.
Okay.
Okay, thanks.
That's a danger sometimes, running out and buying a package. When I worked for a large company, a brand new vice president was very frustrated that the IT department hadn't gotten something validated, and he never understood the whole process. So he went out and bought an off-the-shelf product that they would have had to start from scratch on. And he suddenly was made to understand that, yeah, that takes a long time to validate these things. That those are much bigger than the ones you're talking about. But yeah, a bit of a danger in grabbing something off the shelf without the IT department kind of backing it up. He ended up incurring a huge restock fee for the company because he had to take it back. But he consulted no one before he went out and bought this mammoth system and paid a fortune for it. So yeah, it happens because people just don't understand what's involved in the system. So the issues you raised are perfect ones. Let's see, we have a diminishing amount of time and a few questions left here and there. Let me check and see if anybody else is submitting any.
I do have a question,
Sure.
and I find kind of in the stats program. We run the shelf life estimation in Minitab, but I'm looking for more stat information. What resources would you give to somebody that's been through college years ago and now is in charge of helping run, lead a team that's doing stats, and I need to understand more the models and why we would choose one or the other? Is there Cliff Notes resources out there available?
That's a good question. There are some contractors that teach courses on those that are good. Also, at some of the seminars, like the Kennex one coming up, or some of the mini ones that they've done in the fall, they pull a lot of really great pharmaceutical statisticians in, and their presentations are good, but even better is picking their brain in between the sessions to do that, if you have that opportunity to do that. But otherwise, I think I'd try and get somebody in-house to teach. When I was out at Gilead, we had an instructor that came in and taught my entire group. I was busy that day, but I'm not that great at statistics anyhow, but everybody raved about the amount of knowledge they picked up, and they were quite confident when they were all done. So if you can find the right contractor to do that, or if you're on the verge of going to a conference with statisticians, those are good sources as well. Anybody else have something more specific around statistics that they can recommend?
I was just going to say the vendors themselves often have a lot of YouTubes and things to watch, very specific to their product, but they're helpful as well.
It's a topic that comes up a lot, especially if somebody comes through an inspection or pre-approval inspection, especially, and says, "Okay, so what are you doing with your statistics?" And they're looking to see that you're applying standard packages and methods to do that. Some companies contract it out. Other ones have a special department that does that, and others rely right on the stability group with somebody with a little bit of expertise. But ultimately, you're the owner in the stability group of what happens with your statistics, and whether you farm it out or have it done in-house, you need to kind of give it your stamp of approval that you've run a quality check on, and it's actually working out that the proper methods are being used, and that any abnormalities are explained.
Yes, that's exactly where I'm at. So I need to know more than just the basic regressions and shelf life estimation. Like, is this the right method we're using? So.
Okay, and if I remember, I will post that on Stability Hub, but if you want to send an email or actually go to Stability Hub and put the question on, we might get more exposure and some people that might pop up with some information.
Okay. Thank you.
You're welcome. We're entering the last 5 minutes here, so before we wrap up, I wanted to check and see if there are more questions. Comment from Michael on the chats that Symmetric, SYMMETRIC, is a training provider that has a 3-day course. So there's a quick answer if you want to investigate Symmetric. Any last questions? Okay, wanted to use the opportunity to thank all of our panel and the people who joined us as well. I see that we had like the tide come in and the tide go out. So we have behind us 2 Stability Day celebrations where we took questions as kind of part of the program. This is the first one where we've given the entire program over to questions. So we're looking for feedback, both from our panelists and from the people who attended. Would we do better not to end at 1 o'clock and start at 11? Would we better to grab like 15 minutes after an hour and 15 minutes before? Would you like to see longer sessions with more questions in advance that you can submit to us? We're basically trying to mimic a piece of the old PSDG meetings and see how that flew, whether or not we need to expand into longer sessions or keep it short. Anything along those lines, we're certainly looking for input. So the people who left will try and track down again and take a bit of a poll for them, a survey to see how they liked it, what they might have for suggestions, especially for you panelists that have joined us for the first time. If you have suggestions, we certainly appreciate the people who have been practitioners as well as the suppliers. So if there's anything we can do that helps you from the supplier point of view in getting more information or having people dig a little bit deeper into your area of the industry, we'd be glad to do that as well. So I'm not seeing any final questions. I see a lot of notes of appreciation. Oh, Mike Skidmore, you probably saw this, Sandy, but $800 likely a good price for 8 hours. Of course, that does sound like a bargain to me as well. And he's listed a USP free course as well. So we'll grab all that from our notes and do a bit of a summary for this as well for any key learnings to do that. So let me just say we really appreciated you all joining us, and we're hoping to do it again in October and probably tweak it a little bit more and see where we come out. So thank you all for joining us and participating.
Yeah. Thanks, John.
you.
Thanks, Walter.
Thank you.
Yep.
You're welcome.
Bye-bye.
Good
We'll see ya.
day,
Thank
everyone.
you very much.
Bye-bye.
Bye.
Bye.